Okay, so in chapter 17, we're going to talk about the special senses. So special senses are smell, taste, sight, hearing, and balance. And the reason why we call them special senses, that's because their receptors are located in very specific places. The general senses like pain or touch or vibration, those receptors are found all over your body. So those would be general senses. These are special senses. So we are going to start with the sense of smell, and the sense of smell is called olfaction. Olfaction. And so we're going to talk about the pathway and the receptors in smell. So we'll start by looking at the receptors. So this is the nasal cavity in here. And inside the nasal cavity, you'll recognize the concha in here. They're lined by a mucous membrane. And so the mucous membrane secretes mucus. And so you have a rose, and the chemicals that are coming off of the rose go into the nasal cavity through the nostrils. The mucus breaks those chemicals down, and then those smaller chemicals are able to stimulate the receptors. So here's the olfactory nerve, and the chemicals are going to activate or generate an action potential in the olfactory nerve. Okay? Then that goes to the olfactory bulb. The olfactory bulb leads into the olfactory tract, and the olfactory tract is going to go straight back to the cortex. It does not have to stop in the thalamus. It does not have to synapse on another neuron. It goes right back to the olfactory cortex. And the olfactory cortex is in the temporal lobe of the cerebrum. Okay, so you need to know the path. We go from the olfactory nerve to the olfactory bulb to the olfactory tract, back to the olfactory cortex that's in the temporal lobe. Right? This is showing a picture of what the receptors look like. So the receptors here's the cells. Okay, there's the axon. All those axons of that neuron are going to merge together to form they're going to merge into that olfactory bulb. Okay, but here's the cell body. Here's the dendrite. Here's the dendritic. These are little cilia coming off of the cell body. These are the chemicals, and the chemicals then are binding to those cilia, those receptors there, and that's activating that cell, which will then generate an action potential in those neurons that merge together to form that olfactory nerve. Okay? So now with olfaction, with the sense of smell, I mean, you can smell over 2,000 different smells. And one of the things that we want to know about olfaction is that, you really can't discern what that smell is until the action potential gets to the cortex. So it gets to that olfactory cortex. And then you interpret then, you perceive what that smell is. Now, there's a thing called central adaptation. Central adaptation is when your brain gets a constant stimulus, and then it stops alerting you to the smell, right? So you know that if you walk into like a barn and you can smell that right away, then after a while that smell goes away, right? It's not because the smell goes away, it's because your brain adapted to that. Same thing if you walked into a bakery. You'd smell it at first, but if it's a constant stimulation, your central nervous system, your brain will adapt to that, and you'll stop smelling that then. Okay, so that's central adaptation. The next, and so these, this diagram will be on your lab exam. Okay, you're going to have this on your lab exam. You'd have to be able to identify the olfactory nerve fibers, I believe the olfactory bulb and the olfactory tract. 17.1a. Okay, with taste, of course, we taste things in the mouth, primarily on the tongue. And there's 4 basic tastes: sour, bitter, salty, and sweet. And those taste buds are going to be found throughout the tongue. Okay? They're on the top of the tongue. There's also these other receptors here that are called umami. Umami is a special receptor that's sensitive to an amino acid called glutamate. Glutamate we find in Parmesan cheese, and it's also in chicken broth. So those tastes don't really, they're kind of salty, but they don't really fit into these other ones. So that's the other receptors. The umami receptors will detect that. Right? Then there's receptors in the throat, in the pharynx, and those are water receptors, so they detect the taste of water. Okay? Now, as far as the pathway to go back, we know that already. We studied the cranial nerves. So the anterior 2/3 of the tongue, anything you taste there, that's going to go back to the brain by the facial nerve. The posterior 2/3 of the tongue, whatever you taste back there, that's going to go back to the brain by the glossopharyngeal nerve. Whatever you taste in your throat, that's going to go back to the brain by the vagus nerve. So if we needed to know the 3 cranial nerves that detect taste, it was going to be facial, glossopharyngeal, and vagus. 7, 9, and 10. Those are the 3 nerves that detect taste, right?
Can you say where they were again? Like facial.
So facial is in the anterior 2/3, glossopharyngeal, posterior 1/3, vagus in the throat. Mm-hmm. Now, from there, those nerves, those cranial nerves, are going to go right to the thalamus, because remember, the thalamus is our final relay station. And then they are going to synapse onto another neuron. Okay, they're going to synapse onto other neurons, and then those neurons are going to go to the cerebral cortex. And the area in the cerebral cortex is called the gustatory cortex. We talked about that in chapter 14. The gustatory cortex. The gustatory cortex is in the frontal lobe. Remember when we saw that it pulled apart that lateral sulcus, and we could see the gustatory cortex in the frontal lobe. So that's the pathway. Cranial nerves 7, 9, and 10 go to the thalamus, and then the other neuron that it synapses on, that's going to go to the frontal lobe, the gustatory cortex. Gustatory cortex. Okay. All right. Now, the little bumps that you see on the tongue, those are not the taste buds. Those are little projections of the epithelium, and we call that papilla. Papilla for one, papillae for plural. So those are little papillae. If we look at the papillae, there's 4 different types of papillae. I know there's another one called the foliate papillae. We're going to concentrate on these 3. The circumvallate papilla, the fungiform papilla, and the filiform papilla. The filiform papilla doesn't have any taste buds on it. The fungiform papilla has 4 to 5 taste buds on it. And the circumvallate papilla can have up to 100 taste buds on it. Okay? So this will be a diagram on your lab exam. And you'd have to be able to identify the papilla. And then for lecture, the filiform papilla, since it doesn't have any taste buds on it, that's just for friction. So your tongue's not so slippery, right? So it helps to create a little bit of friction with the food and the saliva and the drink in your mouth. And then, like I said, fungiform has 4 to 5. Circumvallate has has up to 100. All right, so let's look at what a taste bud looks like. Down here at the bottom, this is one taste bud right here. And the one taste bud, and so this would be one taste bud on one of those papillae. And so the taste bud has several cells in it, and these cells are called gustatory cells, these pink cells. And the gustatory cells have these little microvilli on the one end. We call those taste hairs. And all of those taste hairs poke out of a hole in the papilla. And that hole is called the taste pore. So these little microvilli are they're sticking out of this little taste pore. When you bring a chemical into your mouth, whether it's a food or a drink or whatever it is, it stimulates those microvilli. Okay? And then that creates an action potential in that gustatory cell. The axons of all the gustatory cells are going to merge together and form one of those three cranial nerves, 7, 9, or 10, okay, depending on where it is on the tongue. There are also these little basal cells. Basal cells are stem cells, and the stem cells are going to go through mitosis and replace the gustatory cells, because the gustatory cells die off very fast, right? They don't last. They're like a week and they die. So we have to replace those constantly. Okay. All right, so that's taste. All right, these next two, sight and hearing and balance, these next three, we're going to talk about a little more in depth. So I'm going to go through sight, we'll have a break, and we'll come back and we'll look at hearing, all right? So with sight, the first thing we want to do is look at structures. There's a lot of structures. We have the external structures that you can see just by looking at someone's eyes. First, we start with the eyelids. The eyelids are called the palpebrae. There's an upper and a lower palpebrae. Where they come together in the corners, those are called canthus. So we've got a lateral canthus, that's where the eyelids come together on the outside. And then you have a medial canthus, that's where the eyelids come together on the inside. Right inside that medial canthus is that pink fleshy skin, that's called the lacrimal caruncle. And the lacrimal caruncle produces a mucus that's your sleepy head. When you wake up in the morning and you get that mucus inside your eye, that's produced from the lacrimal caruncle. There's some other little glands. If you look on the inside of the eyelid, you'll see some little holes in there. Those are ducts leading into these other glands that are called tarsal glands. Tarsal glands secrete a watery, thin liquid that keeps your eyelids from sticking together. All right, then we look at the eyeball itself, and we can see the white of the eye, that's called the sclera. And then we can see the colored portion of the eye, that's called the iris. And then the black area, that's actually a hole leading into your eye, that's called the pupil. Now you see a sheen right there over the eye. There is a membrane over the iris and the pupil that's called the cornea. So that's why you're seeing this reflection of the cornea. If we take off some of the skin and we look a little bit deeper into the eye, now we can see some other structures. So we can see this gland right here. This is called the lacrimal gland, and that produces tears. So the tears are going to flow through these ducts, and then the tears flow across the eye. They're going to flow through these little canals and into this area that's called the lacrimal sac. And then the lacrimal sac drains into the nasolacrimal duct, and that drains into your nasal cavity. So whenever you're tearing, whenever your eyes are tearing, that's why you start to sniffle, because the tears are draining down into your sinus cavity. And the tears have a little bit of an antimicrobial, they're a little bit of salty, and so they're able to help fight off infection around the eye. All right, so those structures we need to know. Then we can also see that attached to the sclera are these muscles. These muscles are the extraocular muscles. They're skeletal muscles, and they move the eyeball. And we know what cranial nerves move the eyeball, right? 3, 4, and 6, right? Oculomotor, abducens, and trochlear. 3, 4, and 6 move the eyeball. Okay? So we have to name the muscles. And 4 of the muscles are called rectus. Rectus means straight. So they go from the eyeball straight back. Okay? The one on top that goes straight back, that's the superior rectus. The one on the bottom that goes straight back, that's the inferior rectus. We can't see the medial rectus on this picture, but you will be able to on your model. You also can't see the lateral rectus on this model, on this picture, but you would be able to on the model. They go right from the eyeball straight back, okay, on the medial and lateral side. Then we have 2 oblique muscles. So the rectus muscles, if the superior oblique muscle were to contract, it would move your eyeball up. If the inferior rectus would contract, it would move your eyeball down. Right? Lateral rectus moves your eyeball out. Medial rectus moves your eyeball in. Okay? Then we have these oblique muscles because the eyeball can also move at an angle. Right? So we've got the superior oblique muscle right here. So this muscle, its tendon hooks around and then will attach to the sclera. So it's coming around at an oblique angle. That's the superior oblique muscle. Then we have the inferior oblique muscle right here, and that attaches, you can see it coming around at an angle and attaches onto the lateral side of the eye. That's the inferior oblique muscle. And those are extraocular muscles. All right. Okay, so this diagram shows the different layers of the eye. There's 3 layers of the eye. We used to call them tunics, but now we're calling them layers. So the outermost layer is the fibrous layer. You can see that all the way around. The middle layer that's more tan, that's called the vascular layer. And then the innermost layer of the eyeball, that is called the neural layer. So we've got the fibrous layer, the vascular layer, and the neural layer. Okay, so we're going to go through each of the layers and the different structures in each of the layers. So we'll start with the fibrous layer, that's the outside. So the fibrous layer is made up of the sclera, that's the white of your eye, and then that's continuous with the cornea. That's that clear membrane over your iris. Those 2 structures make up the fibrous layer. The sclera, that is a tough, durable tissue that helps to, protect your eye, and it's also where those muscles attach. Those extraocular muscles attach. Okay, it's nice and strong. The cornea also protects the inner eye. But it also helps to bend light. It's completely clear. So when light hits the cornea, it bends it just a little bit so that it will go through the pupil. Okay? So we want that light to go through that hole, that pupil. So that's what the cornea does. Right? So that's your fibrous layer, made up of the cornea and sclera. The next layer, the middle layer, is called the vascular layer. And the vascular layer has 3 structures in it. The iris, which is the colored part of your eye. The ciliary, which is right here. The ciliary body, which is right behind it. Okay, that's also a muscle. So the iris and the ciliary body are both muscles. And then we have the choroid. And the choroid is filled with blood vessels. So it's a very vascular layer. That's why we call it the vascular layer. It supplies the eye with nutrients and oxygen, and it carries away waste products. That's what those blood vessels do. All right, now we're going to look at those 2 muscles. We're going to look at the iris and we're going to look at the ciliary body, and we're going to see what they do. Make a long story short, the iris, that muscle controls the size of the pupil. Okay? The ciliary body, that controls the shape of this structure, which is called the lens. Right? So we'll look at that. Now, before we do that, let me explain some of these other structures that are in there so that you can see. So this is the lens right here. That's the lens. And the lens is attached to the ciliary body by these suspensory ligaments. Okay, so we got suspensory ligaments and the lens. Now, what happens is the lens is really dividing the inside of the eye here into two major cavities. This cavity that's in front of the lens, that's called the anterior cavity. And then this cavity back here that's behind the lens, that's called the posterior cavity. The anterior cavity is filled with a fluid, and that fluid is called the aqueous fluid, aqueous humor. Okay? So aqueous humor, it's a real watery fluid. It keeps the cornea so that it's not sunken in, so it keeps the shape of the cornea. But the doctor, when you go into the eye doctor and they do that poof of air on the cornea, they're checking to see how much fluid is behind that cornea, how much pressure is being placed on that cornea. And if there's increased fluid in there, because the fluid's not draining properly, then that's glaucoma, because there's too much pressure against that cornea. So that's what glaucoma is, too much pressure against the cornea there. So that's a watery fluid. Now in the posterior cavity back here, this is where we have what's called the vitreous humor or the vitreous body. And this is a jelly-like substance that helps to keep the shape of the eye. It helps to keep it round. Those of you that did the dissection, you saw that. It was a clear jelly-like substance, right? So that just packs inside there and keeps that shape of that eyeball. Okay, so now let's go back and let's look at the iris and the ciliary body. Okay, so we'll start with the iris. So the iris has two different muscle orientations. Two different muscles, really, in the iris. This is showing you the two different muscles. It's a smooth muscle. It's involuntary. You really have no control over that muscle. The inner muscle is arranged. It has a sphincter orientation. And then the outer muscle has more of a radial orientation, where the muscle fibers looks like they're radiating away like the spokes of a wheel, right? So the inner muscle here, we call this the pupillary constrictor muscle, because we know when a sphincter muscle contracts, it does this. And what's going to happen to the size of the pupil? It's going to get smaller, right? So that's why that's a pupillary constrictor muscle. So the question might be, what orientation is the muscle, is the pupillary constrictor muscle? And then the answer would be sphincter, right? It has a sphincter orientation. The muscle on the outside here, this is the pupillary dilator muscle. The outside stays fixed, and when the muscle contracts, it pulls in. It shortens this way. So when it shortens this way, what's going to happen to the pupil? It's going to get bigger, and we call that dilation, pupil dilation. Right? So we know that the oculomotor nerve, right? The oculomotor nerve controls the pupillary constrictor muscle, right? So when the oculomotor nerve is stimulating that constrictor muscles, the pupil gets small. Okay, so this is a picture of pupil dilation. So that's when the dilator muscles are pulling and shortening these muscle fibers here. And then this is when the sphincter, the pupillary constrictor muscles, are contracting and making the pupil really small. Okay? So that's the iris. Now the iris is a muscle, but it's also pigmented. So it, everybody has a different eye color. Blue, green, brown, mixture of all of those, because of the pigments in the eye, in the pupil. In the iris. Okay, next thing we're going to do is we're going to look at the neural layer. Okay, the neural layer, and we're going to concentrate on looking at the retina. Okay, so let's look at this picture first. So we see the retina is the orange color here, right? And so this whole area is filled with photoreceptors. Photoreceptors detect light. So once light gets to your back of the eye, it stimulates those photoreceptors. That sends a message to your brain telling you that you see something. Okay? Now, you can see that it's all very smooth until we get to this part right here, and there's a little divot in there. That little divot is called the macula. The macula or the macula lutea. Okay? Macula. And the very center of that macula is called the fovea centralis. So we've got this macula lutea, and then we have the fovea centralis. Okay? That's where there's a high concentration of photoreceptors, especially photoreceptors that are called cones. That's where you have the highest concentration of cones, is right in that macula lutea. Okay? The other thing that we see that's breaking up this retina is this right here. This is the optic nerve, right? So the optic nerve is extending from the eyeball, and so where we see the neurons right here, you can tell that there's no retina right there. There's no photoreceptors right there. So that is called your blind spot or the optic disc. This area inside the eye where there are no photoreceptors is called the optic disc. Okay? And then the optic nerve are the neurons leading away. All right, so what happens? Let's talk about the vision and the optic disc. When light hits the cornea, so you're looking at something, light's hitting the cornea bends the light a little bit to make it pass through the pupil. And then it's going to hit the ciliary body here, and then it's going to hit the lens here, and the lens is going to bend that light even more, and then that light is going to end up right here at the fovea centralis. Right? And I just realized I forgot to talk about the ciliary body, so I've got to go back and tell you how we change the shape of the lens. But let me finish with this first. So the light bends, and then it bends, and we're directing it right back to the fovea. So when you're looking at something directly, that is going right back to your fovea. When you're seeing things in your peripheral vision, that's because that light is hitting photoreceptors somewhere else and not right there in that fovea. Right? So that's what your peripheral vision is. All right, so if you are looking at something and the light is hitting the optic disc, that's your blind spot, and you can't see that. So let me show you this picture here. Okay, so in the second row, actually, Marianne, you guys and Alexis, you in the first row, cover your right eye and look directly at this plus sign. Okay, cover your right eye, look at the plus sign. While you're looking at the plus sign, can you see the circle? You can. In the second row, you guys do it. Look at the plus sign. Can you see the circle? Don't look at the circle. Look at the plus sign. Can you see the circle? Third row. Let's try it. Can you see the circle? You can. So the blind spot would be if you would cover your eye and looking at the plus sign, you would move to a spot where you would no longer be able to see that circle. and that's your blind spot. Okay, that means that the lights coming in your peripheral vision are hitting the that area where the optic disc is, where there are no photoreceptors. Normally we don't do this on a big, overhead. It's a little card, and you move the card forward and back until you see where that blind spot is. That means the light in your peripheral vision is hitting that optic disc. Okay, so that's what the blind spot is. All right, now let's go back and let's look at the Got to take a look at the lens, the ciliary body, right? So there's the ciliary body, and there are these suspensory ligaments that are suspending the lens right there. So this lens is made up of a bunch of little crystals. It's soft. I know when you do the dissection, it's a real hard crystal. That's because the cells have died. But it's made of these cells that are, it's a little more fluid. And so this ciliary body, it can go round or it can go flat. Okay? So if we pulled on the sides of the lens, it would go flat, right? And then if we relaxed, and it would go round again. Now the lens, so the suspensory ligaments, if they pull on the lens, it goes flat. If they relax, it goes round. There's this thing called accommodation. When you're looking at something up close, that's accommodation, and the lens has to go round. The suspensory ligaments would have to relax, and the lens would have to be round in order for the light that's coming in to bend enough to get to that fovea. Right? So the lens would go round. If you're looking far away, those suspensory ligaments are going to pull tight, and that lens is going to get flat so that it doesn't have to bend as much, and it'll make its way right back there to that fovea. Okay? So that's the lens. Now, the ciliary body controls whether those ligaments are pulling or relaxing. Okay? Now, the muscle itself is round. So here's your lens. And when you see the model, you're going to see this, but there's the lens, and then around that, this whole thing, okay, this is your ciliary body, which is a muscle, and this is the lens, okay, and then you have suspensory ligaments attaching the lens to the ciliary body, right? All right, so if you think of this kind of like a dream catcher, you got a dream catcher and you've got all those strings inside. If this muscle were to contract, what would happen to the suspensory ligaments? Would they be tight or would they be loose? Loose, right? So when the muscle contracts, the ligaments go loose and the lens goes round. So it's kind of opposite of what you would think, right? And then what happens when the ciliary body relaxes? So the sphincter muscle relaxes. What happens to the ligaments? They tighten. What happens to the lens? It gets flat, and then you're looking far away. That's what happens when you're looking far away. So that's going to be a question on the exam. What happens? What happens to the muscle, the suspensory ligament, and the lens when you're looking up close or far away? Right? Okay. So that's the ciliary body. Couple other. Let's look at the photoreceptors in the retina. Okay, so this is a layer. This is the retina. So this is the inside of the eyeball where that vitreous body would be. Out there would be the choroid and then the sclera. And so light's coming in through the eye, and it has to pass through these different cells. These cells up here are the photoreceptors. There's two different types of photoreceptors. There's cones and there's rods. There's a lot more rods than there are cones. The rods help you to see in dim light. They're not really trying to detect anything, any detail. They're not trying to detect any they're trying to do is detect anything in dim light. So, you know, when the lights are dimmed, you're not going to really see sharp borders on anything. You're not going to see color, right? Okay. The dimmer it gets, the more difficult it is to see borders, the more difficult it is to see colors, right? And then the more light there is, then the cones become stimulated. So once we have brighter light, now the cones become stimulated. The cones help you to see detail, borders, outlines, colors. Okay, so the receptors then, once they're activated, whether it's the cones are activated or the rods are activated, they're going to start an action potential in these neurons. These neurons are bipolar neurons. We call them bipolar cells. Do you remember the bipolar neuron when we talked about multipolar, unipolar, bipolar, annexonic? That means this, you can see it looks like a bipolar neuron. It has a dendrite on one side and axon on the other side, right? It's a short little neuron. So this one generates an action potential, and then the bipolar neuron starts an action potential in the next neuron. The next neuron is called a ganglion cell. Right? So we had the photoreceptors that activated the bipolar cells. The bipolar cells activated the ganglion cells. The ganglion cells have axons, and all of those axons are going to merge together to form that optic nerve. Right? So, interesting, before I show you the next slide, here's the deal. It's so kind of weird. The light comes into the back of the eye, right? It comes into the back of the eye, and it has to work its way through the ganglion cells, and then it has to work its way through the bipolar cells, and then finally it hits those photoreceptors. Then the photoreceptors become activated, then the bipolar cells generate an action potential, then the ganglion cells generate an action potential, and those axons merge together to form the optic nerve. It's just kind of weird. Why wouldn't the photoreceptors be here? You know, so the light actually becomes a little filtered as it works its way through those cells, in between those cells, to get to the photoreceptors. Okay? So the photoreceptors are more on the outside. All right, let's talk a little bit about the cones then. The cones, there's three types of cones, three types of cells that can detect color and light. Actually, here's another picture. Let me show you this first. This is the optic nerve, right? So the optic nerve contains the axons of all those ganglion cells. Here's the ganglion cells in this layer. So here's all the neurons coming together to form that optic nerve. This is the optic disc because there are no photoreceptors in this area. Okay? Here's the bipolar cells. Here's the photoreceptors. So these are the rods and the cones. Light comes in, it filters through, activates the photoreceptor, generates an action potential in the bipolar, generates an action potential in the ganglion cells, and then the action potentials travel down the axons through the optic nerve. Okay? All right, so, all right, let's look at the cones. Come back to that one. Okay, there's three different types of cones. There's a blue cone, a green cone, and a red cone. The blue cone is only going to be sensitive to a certain wavelength. So light has wavelengths. Blue cones are going to be stimulated by shorter wavelengths. Okay? Green cones are going to be stimulated by wavelengths that are somewhere in the middle, and red cones are going to be stimulated by longer wavelengths. The human eye can only see somewhere between 400 and 700 nanometers of wavelength, right? And those three cones are, they take up all of that between 400 and 700. Blue are short, red are long. Now, if all three of those cones are stimulated at the same time, you're going to see white. So when you're looking at a fluorescent light, all three of those cones are stimulated and you see white. When you see a white piece of paper, what happens there is that, or anything that's white, you're not looking at light now, but the light's hitting the paper and bouncing back and stimulating all three receptors. On the other hand, if you're looking at something black, right? If you're looking at something black, the light hit the object and absorbed into the black, and now none of your receptors are being activated, and that's why you're seeing black. Okay. And the other colors are anywhere in between. Some of the receptors are being activated, some of them are not. Okay. Pure red would mean that you're, only the red, it would be a wavelength closer to 700 nanometers, and only those red cones would be activated, not red and green. Okay. If it were a little more orange. All right, so that's important. All three cones become activated when you see white. All right, let's go back. I want to show you this picture here. This is going to be on your exam, on your lab exam. So on this picture, you can see this space and this space. This makes up the anterior cavity. And then the iris divides the anterior cavity into two smaller chambers. So in front of the iris, we have the anterior chamber. And then behind the iris, we have the posterior chamber. So the anterior and posterior chamber make up the anterior cavity. Right? So when it's asking for the space, you want to be as specific as possible. So this is the anterior chamber, posterior cavity. Anterior chamber is filled with an aqueous humor. Posterior chamber is filled with that vitreous body or vitreous humor. So the aqueous, this fluid up in here that I talked about, that's going to be produced and then it's going to drain. And when it drains, to keep it fresh. So when it drains, it's going to drain through this opening right there that's called the scleral venous sinus. So that opening and that opening there, that's the scleral venous sinus, and that's also on your lab exam. So this diagram will be on your lab exam. These are the suspensory ligaments. This is the ciliary body. This is the iris. Okay. So you just want to make sure you study that diagram as well. And one last thing. I don't want to forget this. This picture is also going to be on the lab exam because this is the sclera. Covering the sclera is, you can see it looks pink here. This is the conjunctiva. It's actually clear. It's translucent on your eye, so you don't really see it. So it's a translucent tissue that overlying the cornea, or I'm sorry, overlying the sclera, and that's called the ocular conjunctiva. And then it bends around and it covers the inside of the eyelid, and that's called the palpebral conjunctiva. And we see it up there too, on top. We can see the ocular conjunctiva, and it bends around and it becomes the palpebral conjunctiva. Okay, so this picture, there's the ocular, and then there's the palpebral conjunctiva. All right, so this is helping to keep bacteria away from the eye. It's also, because it bends back there, you can never lose anything in the back of your eye. So you can't lose that contact. That contact is never going to go back here by that optic nerve. Dirt, makeup, it's going to pile up in here, but it won't go back to the back of the eye. Okay. All right. Let me just double check, make sure I don't need anything else to be said. There's one other thing, I guess, with eyesight. Oh, a couple things two more things, and then we're going to take a break. The pathway for the eye. We are not going to learn the physiology of the receptors, but as far as the pathway goes, your right eye has a visual field and your left eye has a visual field. And so we don't want the right eye just to tell the right brain what's going on. We want the right eye to tell the right brain and the left brain. We want the left eye to tell the left brain and the right brain, right? So we want to crisscross our messages so we don't have double vision. So whatever you see, all those ganglion neurons, those axons, converge together to form the optic nerve. We have an optic nerve from the left eye, an optic nerve from the right eye, and they meet at that area called the optic chiasm. You guys saw that optic chiasm on the sheep brain dissection. At the optic chiasm, some of the neurons are going to stay on the same side and continue to the thalamus. The other neurons, the other axons are going to cross over and go to the opposite side, okay, to the opposite side of the thalamus, right? So it's going to go to the opposite brain. So that's what it does. We've got neurons, they crisscross or they stay on the same side at the optic chiasm. Then they go to the thalamus and they synapse on another neuron. And then that other neuron is going to go to the visual cortex in what lobe? Occipital lobe, right? So that's what happens. That's the pathway with vision, right? So we've got the pathway of vision, and what was the last thing? One other thing. Oh, as far as normal, like normal sight, normal vision. When you always normal vision is 20/20. Okay, that's what we did was we said here's some letters. And a person that has normal vision can read those letters from 20 feet away from it. And those, so that's what we said. So that's 20/20. So if you have 20/20 vision, you're like the normal population that can see something at 20 feet away. If you have better than normal vision, say it's 15, you're 15/20, that means that a person that can see those letters from 15 feet away, you can actually see those letters from 20 feet away. So you have better vision because you can see farther than they can. Okay? A person that has worse than normal vision, like 20, so the other one was 20/15, not 15/20. 20/15 is better than normal. A person that has worse than normal vision would have like 200. That means that that person can see at 200 feet away, you have to move up to 20 feet in order to see, right? So that's the difference. So 20/20 is normal, 20/15 is better than normal, anything 20/30, 20/40, 20/50, 20/100, 20/200, that's worse than normal. A person that is 20/200, even when wearing glasses or contacts, is considered legally blind. Okay. All right, so that's it for the eye. I'm going to give you just like 4 minutes, and then we're going to start on the ear right away, okay, to make sure that we get done with hearing and balance.
So,
Okay, so in the external ear, we have the cartilage and the fleshy part of the ear, and that's called the auricle. And that kind of acts like a funnel, and any airwaves, any sound that's going to be funneled into this canal here, and this canal is called the external acoustic meatus. External acoustic meatus. The external acoustic meatus is surrounded by a mucous membrane that secretes a very specific substance called cerumen. Cerumen is earwax. So earwax helps to prevent the bugs and the dust and bacteria and things from getting into your ear. So that's what the external ear is. Okay? Then we have the middle ear, and the middle ear is made up of the tympanic membrane. That's the eardrum. And then behind the eardrum, we have these little bones. Those are called auditory ossicles. So the auditory ossicles are made up of the incus, the malleus, and the stapes. The stapes is the one that we're going to pay attention to because underneath the stapes is a window into the inner ear. All right. So then we have where those bones are is an area called the tympanic cavity. And the tympanic cavity is it's filled with air. Okay, so we've got air on both sides of the tympanic membrane. Okay. All right, so that's the middle ear. And then in the inner ear, which is going to be everything from that stapes, from right behind the stapes and back, we have this tube right here, which is called the auditory tube. And the auditory tube goes down to the nasopharynx. So the nasopharynx is your nasal cavity and your throat. So when you get a cold or you have a sinus infection or you're all stuffy up in there, some of that bacteria, if you have bacteria or a virus, that can actually travel back up that auditory tube and get into the middle ear, and then you have a middle ear infection. It's called otitis media, right? So that's a middle ear infection. Now we see that a lot in young kids and infants because this auditory tube, which is more vertical in an adult, the structures haven't grown, and that auditory tube will be more horizontal. So then it's a lot easier for the bacteria and the viruses to get into the middle ear going horizontally than it would be trying to fight gravity and go vertically.
Is that where they put like tubes in your ear?
Right, so if they get chronic ear infections, one thing they might do is put little tubes in the eardrum so that the fluid that accumulates in here because of the infection can drain out this way, because it's obviously not draining fast enough this way. Because normally this should be air in there, it should not be fluid. Okay?
Does the tympanus, does it like open a little bit?
No.
No?
Nope, it doesn't open. It doesn't open.
Because when sometimes you can like
Hear it pop?
yeah, but also I never heard that, but sometimes it gets like the fluid from the mucus can come off from the ear.
Only if there's a hole in the tympanic membrane. Sometimes the tympanic membrane can burst if there's fluid, if there's too much fluid pushing against it, and then the fluid will come out.
And it won't
But it has-
restore. It will.
It will.
It will restore.
Right. Did you guys have questions? Did you guys have questions? No? Okay. All right, so what happens with the tubes then is the tubes will just fall out on their own, and that membrane, which is made of cells, will just grow back together and it'll heal back together. Okay? So, all right, so that's the auditory tube. Then we have this other structure in the inner ear, and that's made out of this, it's a bone in there. It's called the bony labyrinth, and it looks like a snail to me, right? This looks like the snail's body. That's what we call the cochlea. This looks like the snail's head. That we call the vestibule. And then these things that look like an antenna coming off of the head, those are called the semicircular ducts or the semicircular canals. Okay. Each of them have different receptors that are going to detect different things. The cochlea, that detects hearing. The vestibule, that detects acceleration, deceleration, and gravity going up and down. It detects when your body's going up and down, and when you're going forward and when you're going backward. Okay? The semicircular canals here, they detect the movement of your head. If you turn your head from to the side, if you nod, if you put your ear down to your shoulder. Those three ducts are going to detect the movement of your head. So what we're going to do now quickly is we're going to go through each one of those areas, and I'm going to show you the receptors. All three of them have what are called hair receptors, so little tiny hairs that detect the movement of fluid against them. Right? So you've got fluid or pressure. They're like mechanoreceptors, and when they bend from either fluid or pressure, that's going to stimulate an action potential. That action potential is going to, those neurons, those axons will converge together into the vestibulocochlear nerve, which goes back to your brain. Okay? All right, so let's take a look at the first ones. So we're first going to look at the semicircular canals. So the semicircular canals, like I said, they detect movement of your head. Now, the outside of this whole thing is made of bone, but on the inside of this whole thing is another labyrinth that's identical to it, but it's made out of a membrane. So we have the bony labyrinth on the outside, and then on the inside is the membranous labyrinth. And so there's a space in between the bony labyrinth. So this blue here, this would be the membranous labyrinth, okay? And it's hollow. And then on the outside is the bony labyrinth. So there's fluid in between the bony labyrinth and the membranous labyrinth, and there's fluid inside the membranous labyrinth. So we name the fluid. The fluid on the outside of the membranous, on the membrane, on the outside of the membrane is called the endolymph, or perilymph. And the fluid inside the membrane, that's called the endolymph. So we just call it perilymph if it's on the outside of the membrane, endolymph if it's on the inside of the membrane. So there's fluid in there. And so we see, there's fluid running through the tubes, there's fluid in the vestibule, and there's fluid in the cochlea. All right, now the semicircular canals. The receptors for those are found inside the membranous labyrinth at the base of the semicircular canal. And so there's these purple things here, those are the receptors. I'm going to show you a bigger picture of it in a second, but they have little hairs that are sticking up. Little hairs. Those are hair receptors. And so when the fluid inside this membranous labyrinth, when that hits those hairs, the hairs bend. And then your neurons will be activated and a message will be sent to your brain telling you which direction your head moved, right? So, for instance, if you nod your head, there's three different semicircular canals. There's an anterior semicircular duct, there's a lateral, and there's a posterior semicircular duct or canal. So if you're nodding your head, you're only moving the fluid in that anterior duct. So you're moving the fluid in the anterior duct. It hits the hair cells. The hair cells generate an action potential. That action potential is going to go back to the brain by the vestibular branch of the vestibulocochlear nerve, and your brain understands then that you are nodding your head. Okay? If you were to shake your head no, then it's going to be the fluid in these lateral ducts that move. That's going to hit the hair cells, and then you generate an action potential that goes to the brain. It tells your brain that you're shaking your head no. If you put your ear down to your shoulders, then you're only moving the fluid in these posterior ducts, okay? And then that's going to press on those, it's going to bend those hair cells. Message goes back to your brain, telling your brain that you're bending your head to the side. So let's take a look at the receptors to see what the receptors look like. Okay, these are the little hair cells. Here's the little hair cells. They have little microvilli sticking out from them. That's what we call hair. Okay, the little microvilli. The little hair cells are embedded in a jelly-type substance in the semicircular duct, and then that's just to keep them standing upright. And then there's fluid on either side. So when you're nodding your head in that anterior duct, the water moves across and bends the hairs. That starts an action potential in the vestibular branch of the vestibulocochlear nerve, and that goes back to your auditory. Goes to the thalamus first, and then to the auditory cortex. Okay. If this were in the lateral duct, it would be the fluid would move if you shook your head. Or in the posterior duct, the fluid would move if you put your head to the side, right? But it bends the hairs, that stimulates or activates the receptor, that starts an action potential in those neurons. Okay? All right, so that's the movement of your head. Now, as far as the movement of acceleration, deceleration, and gravity goes, those receptors are found in the vestibule. So here's the vestibule area in here. There's two areas in the vestibule that are called the utricle and the saccule. The utricle and the saccule. They also have hair cells, very, very similar, okay, to what was in the semicircular canals. But they're going to detect, they're going to be stimulated if you're moving forward or backward, right? And then those hair cells are going to bend. Or if you're going up or down, then those hair cells are going to bend, okay? So that's what it helps to detect that. So this is what the receptors look like. Okay, this is what the receptors look like. Here's the little hair cells. So this is in the utricle and saccule. There's this jelly-like substance above it, and sitting on top of the jelly are these crystals. That's to keep it, to hold it down a little bit, so that there's not the movement doesn't happen. You wouldn't want to always be feeling like you're accelerating and decelerating, so it keeps it just a little more stabilized, right? The crystals are called statoconia. Now, occasionally the statoconia break off, and if they break off, so they're in here in the utricle and the saccule, and if they break off, then the little crystal can travel into one of the semicircular canals and start bending one of those hair cells. And then what happens? Then you feel like your head is moving constantly because there's this constant crystal on your hair cell. And what is that called when you feel like your head is moving constantly? Vertigo. That's vertigo, right? So they have a way, they have these different positions they can put your head in to try to move the crystal away so that your body has time to reabsorb it to get rid of the vertigo. But that's what a benign paroxysmal vertigo is. All right, so then the last thing that we have to look at is hearing. And we have to look at the cochlea for hearing. And I'm going to show you this picture here. So this is the cochlea. Remember the cochlea looked like the body of a snail, and it was all, wound around itself. This is the cochlea kind of unwound. So we just rolled it out so you can see from one end to the other end of the cochlea. So we've got to go over some specific structures in the cochlea, and then we're going to talk about sound. So we have, here's your stapes. Underneath the stapes is an oval window that leads into the cochlea. Here we have the membranous is the border on the outside. The bony labyrinth is on the outside. The membranous labyrinth is on the inside. And so we end up with these three passageways or these three ducts. We have the vestibular duct on top right here. The membranous labyrinth we call the cochlear duct, and then underneath it we have the tympanic duct. So when we're talking about how we hear things, we're going to use those terms. Vestibular duct, cochlear duct, and tympanic duct. Okay. The receptors for hearing are in, we used to call it the organ of Corti, now we call it the spiral organ. They're found in the cochlear duct. So the cochlear duct has a top and a bottom to it. The top is the vestibular membrane. The bottom is the basilar membrane. Right? All right. So let me give you the big picture. Then we're going to break it down a little bit so you can see how this works. So you get sound waves coming in through your external acoustic meatus, and those sound waves hit the tympanic membrane and make that tympanic membrane start to vibrate. That causes the auditory ossicles to vibrate, each one of them. And finally, when the last one vibrates, which is the stapes, that starts to vibrate the oval window. The oval window then starts to produce water waves. So it's producing these waves in the water. There's pressure waves in the water that's in the vestibular duct. So the water in the vestibular duct is called perilymph. So we're getting pressure waves in the perilymph. Okay? That pressure is eventually going to hit the cochlear duct. And when it hits the cochlear duct, it causes the bottom membrane here, called the basilar membrane, to bounce down and then back up. The little hair cells are sitting inside there. So when the basilar membrane bounces down and then bounces back up, it causes that hair cell to bend, right? And that activates, that's then activating that receptor. It's causing that hair cell to bend. Okay? So then that generates an action potential in the cochlear branch of the vestibulocochlear nerve, in those neurons. And that's going to go back to your thalamus, okay? And then back to your cortex. Now, we have to get rid of the pressure. If we don't get rid of the pressure, just because it hit the basilar membrane doesn't mean we got rid of the pressure. We still have pressure waves in the fluid. So we have to get rid of the pressure. So the pressure then follows, it's going to go all the way through that cochlear duct to the tympanic duct, and the pressure waves go all the way out to this window, which is called the round window. And that has kind of like a drum seal over it. So the pressure waves hit it, and it expands, and it kind of, it just absorbs that pressure. That's what it's doing, is absorbing the pressure. Now we got rid of the pressure. Yep.
You're bringing in your ears. What is that?
So ringing in the ears can be caused from several different things. It could be caused from constant stimulation here. Most of the time it's caused by some type of irritation on the cochlear branch of the vestibulocochlear nerve. So now let me show you what the spiral organ looks like. So that's the receptors that are found right here in the cochlear duct. Okay, this is what it looks like. So this is that basilar membrane. Here's the hair cells. Okay, that's in the cochlear duct. The fluid, the perilymph up here, this is receiving, so this is up here where you're going to get those pressure waves. Pressure waves hit, and it causes this basilar membrane to bounce. And when the basilar membrane bounces down and then bounces back, it's going to push these hair cells up against the top membrane and make them bend. Now we've just activated our receptor. That's going to generate an action potential in these neurons. The action potentials travel down the axons. All those axons converge into the cochlear branch of the vestibulocochlear nerve. That goes to the thalamus, synapses on another neuron. That's going to go to the auditory cortex in your cerebrum. Okay? A lot going on, right? There's a lot going on. You have to know the steps of hearing. You have to know these steps. Know the steps of hearing. If you have a high pitch, it's going to hit down closer here. If you have a low pitch, it's going to hit further down. And that's how your brain discerns between high and low pitch. Right? So you have to know that. So are there any questions? A lot to know in chapter 17, right? The important thing right now is to get those lab structures down, right? Because you have your lab exam on Friday. You'll have time over the weekend to study. Next, your Chapter 17 homework quiz, will be due next Wednesday. Okay? All right. We are doing